We have Lab 17, the special senses. We're beginning with gustation, looking at the structure of the tongue, the various papillae. And for this section on the tongue, we're using a textbook image. So that's the figure right here, 18.7. And just taking a look here, it's all labeled. We have the filiform papillae that are at the medial portion of the tongue, so the tip and down the medial section. On the lateral aspect of the tongue, the larger fungiform papillae, and meaning the circular wall here in the back, the circumvallate papillae. So the three shapes all seen on this figure. We're now moving into the section for hearing. We have the structure of the ear, and we'll start with the external ear. So the external ear, meaning always at the surface, the external portion. The middle ear section will change colors, and the middle ear here highlighted in this green region, the middle ear region. And then lastly, there's the area of the inner ear. Again, we'll change to a different color, and that's what we can see right here. So external, of course, at the surface, middle ear, and then the area of the inner ear. So let's start at the external ear, and we'll go through the detailed components. So that's where we see the terms here, the auricle, the tragus, and whatnot. So let's begin there. So we have the model, and we'll start with the auricle. That's essentially just this funnel structure here on the side of our head, or another view of the auricle region. We can see down the canal of the external acoustic meatus. And at the end of that canal, this here is the tympanic membrane. We're just going back to the model. We see the view of the external acoustic meatus here. Not the best view from this angle for the tympanic membrane. And lastly, there's the term tragus. We have to go to this model. The auricle is this region here. And this particular piece of cartilage right in here is the area of the tragus. And that's all the structures for the external ear. So now going on to the middle ear region. Again, we're looking in this area here. And we can see that there's these small little bones here, collectively known as ossicles. And to get a closer look at them, there's the malleus, is the one connected to the tympanic membrane. And the second bone is the incus. And the third bone is the stapes. And here's now a closer view. So here we can actually see that tympanic membrane, and connected to it, the malleus. The next bone over is the incus. And truly shaped like a stirrup, there's the bone of the stapes. And the final part of the middle ear, we have this canal here known as a auditory tube. You notice running there at an angle. So this is showing the adult angled auditory tube on any of these views. So now moving into the area of the inner ear, that's what we're viewing here. And you can see we're looking at this cavity inside of the temporal bone. And for this detail, we're going to start first with this labeled figure. So just to describe the concept, we have what's called the bony labyrinth. And here on this illustration is anywhere we see the tan color. So it's the superficial bony surface of this structure of the inner ear. Whereas the membranous labyrinth on this same figure is internally, and it's shaded in blue. So anywhere and everywhere we see the blue shading on this figure. And we notice the membranous labyrinth is deep to the bony labyrinth. And this is now the entire inner ear region. And we're going to start here with the spiraling segment. This spiraling segment is known as the cochlea. And to take a closer look at the cochlea, we're going to zoom in here, and you notice how this model is designed to open up. And that's what we're going to do, is we're going to split open the cochlea and take a closer look. So that's where we get this view here, a cross section of the cochlea. So within the inner ear, the cross section of the cochlea is what we're viewing here. And we'll go through the chambers. So this chamber here is called a vestibular duct. The one in the center, cochlear duct. And down here is a tympanic duct. They're filled with the different fluids. So you notice the vestibular duct is filled with perilymph fluid. And that's also the case for the tympanic duct, also with perilymph fluid, whereas the cochlear duct contains endolymph fluid. With the three spaces, or the duct structures, there's now the membranes coming across. So there's a vestibular membrane. This separates vestibular duct from cochlear duct. Within the cochlear duct, this incomplete membrane, it doesn't go all the way across, is the tectorial membrane. And the tectorial membrane itself is involved in this larger area I'll box in. So everything here in the box is called organ of Corti. And this whole bulging section you see here is resting right on this membrane here, this kind of grayish line. That is a basilar membrane. The basilar membrane separates the cochlear duct from the tympanic duct. And again, all of this view here is a cross section through the spiraling cochlea to see that internal feature. So again, we'll go back to this figure. All of the external tan surface on this figure is bony labyrinth. All of the internal blue shading is the membranous labyrinth all throughout. And we have a model that shows this detail you notice here. So we have the external portion here in white as the bony labyrinth of the inner ear. And then we can see a portion of the internal area in the gray shades. And that's all the area of the membranous labyrinth, the entire gray shaded area. So now we're moving again through the lab down to the section for equilibrium, which is still a inner ear function. So now we have some specific terminology here. The semicircular canals, that consists only of the three looping areas of the bony labyrinth. The same goes for the semicircular duct. They're only the three looping areas of the membranous labyrinth. So again, let's bring up the figures first. So again, the entire tan area everywhere at the surface of this image is all bony labyrinth. However, when we look specifically just at these three loops, the specific name for the external portion of these three loops are semicircular canals. Whereas now, the deep internal portion of these three loops, specifically just the three looping areas, the internal portion, the semicircular ducts are now just a small portion of the larger membranous labyrinth. We'll finish up with the terms of the ampullae. You notice at the base of each semicircular duct, it expands to form an ampulla. The next bulge over is the utricle, and then the bulge closest to the spiraling cochlea is the saccule. So the ampullae close to the end of the semicircular duct, utricle, and then saccule in that order. And then lastly, we have the terminology of the windows, and we'll just point out their placement. So we can see here where the ossicle, specifically the stapes, connects on to the internal ear region. And it's right at that location here, we could see truly that oval shape. This is where the stapes bone would attach, and this is appropriately called the oval window. Whereas here, this is known as the round window. So two features on the area of the inner ear. Oval window is oval shape, and then we have the round window. So now moving on to the section on vision. So starting with the eyes, we have the various tunic layers from superficial, fibrous tunic, the middle layer, vascular tunic, and the deepest layer, the nervous tunic. And just going across the page, we see the components of each of these three tunics. So just going across the page. So let's begin there. And we can start with a image from the textbook. So to begin with the fibrous tunic, the superficial surface of the eye, in the anterior portion, there's the clear cornea, as well as the majority is made of the white sclera. The middle layer, known as vascular tunic, and a number of components. Here in the more posterior portion of the eye, this middle layer specifically is made of the choroid, rich in blood vessels. Whereas we go to the anterior portion, all of this area here and wrapping around, that's known as a ciliary body. The iris, of course, that area that's, of course, going to dictate the color of our eye, specifically the pigmentation of our iris. And then now deeper in, the nervous or neural tunic of the eye. And again, starting in the posterior side, the deepest layer, the structure we see is the retina. And now on the retina specifically, you notice right through the center of the eye, there's this area here, this depression, known as a fovea. And where we see the connection to the optic nerve, where the retina connects to the optic nerve, this is the area of the optic disc. So now going back to the lab, the eye is filled with these two different types of fluids, the aqueous humor and the vitreous humor, and they fill these two different spaces, an anterior cavity and a posterior cavity. So describe these in relationship to the lens of the eye. So let's begin there. We see here the lens of the eye. All of the space anterior to the lens, that is filled with aqueous humor. So truly between the cornea and the lens, all of that space is called anterior cavity. And the entire anterior cavity, made of these two different chambers, together make anterior cavity. And the anterior cavity is filled with aqueous humor. All of the space inside the eye posterior to the lens, much larger space, is known as a posterior cavity, that's filled with the vitreous humor, or it can be called vitreous body. So to see the models and the chart, we'll go through some of those same terms. So again, the tunics are the major layers of the eye. So starting with the fibrous tunic, there's both the sclera and the cornea together making the fibrous tunic. Next, in the vascular tunic, there's the area of the choroid with the blood vessels. We can see the iris. So features from this area. Not the best view for the ciliary body. We can see the lens. And connecting the ciliary body to the lens, here illustrated as these white lines, that's the term suspensory ligaments. And then now the nervous tunic, that's what we're seeing here specifically is the retina. And cutting through that, we're just looking into the space of the posterior cavity that is filled with the vitreous humor. So vitreous humor is fluid, the liquid. Posterior cavity is the space. And a view of the model. So we can see the white sclera. Here we're seeing the area of the iris. The cornea has been removed. We can see a portion of the ciliary body. So that means back here, we're looking at the area of the choroid. And then the internal kind of tan surface would be the retina. And on the retina, we're able to see the labeling for a fovea. And you can see the nerve coming out. So this must be the area of the optic disc. So again, these suspensory ligaments we pointed to earlier, these are the ligaments, the connections between the ciliary body and the lens. So let's see that once more, suspensory ligaments. First from this labeled image, we can see it labeled down here. So between the ciliary body and the lens, those connecting suspensory ligaments. The little hole that I'm pointing to right here, it's not labeled on the image. That's the term canal of Schlemm. So that small little hole that we see here, just about where the cornea and the sclera meet, is a canal of Schlemm. Palpebrae, this is just the anatomical term for our eyelids. Eyelids are the palpebrae. And now surrounding the eye, on the superior lateral side, the lacrimal gland, the tear-producing gland. And let's just go right here. So on the superior lateral aspect of the eye, it's not truly connected to the eye. It's a location of the lacrimal gland. And on the medial aspect of the eye, this is where we see the nasolacrimal punctum. So we can see here, that's the location of the lacrimal gland. We can see the skin and of course the muscle that forms the palpebrae. And now we have the term, the lacrimal punctum, are the little pores, specifically right here, little holes. That's the term of the lacrimal punctum. We produce tears from the lacrimal gland. All palpebrae wash the tears medially, and most of our tears would drain into the lacrimal punctum, down the nasolacrimal duct, and into our nasal cavity. So we have the various models and posters that we'll use for this section. So of course here we just see just the fibrous tunic, sclera, the cornea. Since the cornea is transparent, we can see internally into the iris. We went through this chart, it has multiple features to see from this image. Same for this model here, just opening up the eye. Here again, the lacrimal gland. And again, we just see the sclera. The cornea is clear, and in this case, we see a blue iris. Opening up this model, we can now see, of course, the area of the lens right here, and representing the space and the fluid within. The posterior cavity is the space posterior to the lens, and the fluid contained would be the vitreous humor. And now with that section removed, we're able to see, of course, the structure of the ciliary body. And again, we've already went through the lacrimal gland, lacrimal punctum, and the eyelids called palpebrae. So we'll move on now to the histology, and we're looking at, once again, the cochlea and cross-section. And all of this cochlea histology will be just like the model that we covered for this section. All of those same terms. So let's just go to the tissue sample. So reviewing this here, and we'll just go over the labeling. So once again, where you see the bulging section in the middle chamber is the cochlear duct. And again, that area of the bulge that's protruding here is all this area of the organa corti. The organa corti itself is made of this incomplete extension. Right across here is the tectorial membrane. And it's sitting right on this membrane here known as the basilar membrane. So always on the opposite end of the organa corti is protruding into the cochlear duct. So on the opposite side, this has to be the tympanic duct. So now we have tympanic duct bordered by the basilar membrane, the whole bulging section of the organa corti, and that specific flap of tissue, the tectorial membrane. Let me put that correctly, tectorial membrane. The space in the middle is the cochlear duct. So then the last two features, this extremely thin, delicate membrane is vestibular membrane, and then lastly, a vestibular duct. So you could go back and compare this to that model, and again, it's the same features that we're seeing here from the histology. Moving on to the tissue sample for the eye, and specifically we're looking at the posterior portion of the eye. So it's out here, this first very thick layer. We're seeing the staining, all the strong collagen in the sclera. Going deeper in, this really dense dark section is the choroid. And now the remainder of what we see all the way across to here, this is all the retina as a whole. And now the details of the retina. We have the pink and the purple of course staining of nuclei. So it's now this area here is where we're seeing the photoreceptors. So whether they're rods or cones. The next layer here is where we're seeing bipolar cells, meaning bipolar neurons. And then the third really sparse row you notice, so far fewer nuclei. This layer here is where we have the ganglion cells. So it's ganglion, bipolar, photoreceptors. Collectively, all of that is seen within the retina. And the final sample for special senses, looking at the tongue, our gustatory organ for taste. And we have the term the papillae. That's this entire bulging segment. So here we're boxing in just one papilla, and we can see a number of them here on the screen. So now we'll just zoom in closer. Here we are. We have one large zoomed in papilla here in the image. And on the lateral portions, this entire cluster here is a taste bud. And we can see multiple taste buds, of course, on the edge, on the lateral border of the papillae, taste buds. And we can see the staining of the cells within. And the cells within, that's where we're labeling our gustatory receptor cells. We'll go into laboratory 18, the endocrine system. So we have all of these features here seen on a single model. And that's what we have here. So we could point out the thyroid gland. You notice it's here wrapped around the area of the larynx. That's the thyroid gland. We have the adrenal gland seen superior to the kidneys. Adrenal glands, or another name, suprarenal glands. We have the pancreas. And then lastly, here you notice the uterus fallopian tubes. So this is showing the female gonad placement to ovary. And then down here, the male gonad placement differences. So all of this section here would be the darker stained anterior pituitary. And now you notice the lighter staining. This is the area of the posterior pituitary. So it'd be represented like so. It's basically just front and back, anterior, posterior. So half of the view is dark, half of the view is lighter for the posterior pituitary. The pancreas, again, sticking with the same color scheme, and that's what we see here is the pancreas. You notice all of the darker surrounding tissue, but we're now focused on just now this small light stained, and when I mean light, the color of the cytoplasm, not specifically the nucleus. So we see the darker staining, but this lighter stained cluster here, this is known as a pancreatic islet. So here you notice we're seeing this pancreatic islet, the lighter stained cells, and on all sides, completely surrounded by the darker staining majority. And then lastly, the adrenal gland. Here we're seeing the structure of the adrenal gland. And you can see now here, wrapping around the superficial part of the organ with the lighter staining. That is the adrenal cortex. And now the deeper internal portion with the darker stain to it. This is all the area of the adrenal medulla. So medulla is the deeper portion of some organ. The cortex is the superficial portion. And we see the different color treatments there.